Hello and welcome to today's Cannabis and Hemp Research Forum, hosted by the UC Davis Cannabis and Hemp Research Center. I am Dr. Cameron Carter, and along with Li Tian, we are the co-directors of the center. The purpose of this monthly forum is to inform our campus community, members of the cannabis and hemp industry, and the general public about cannabis and hemp research that's happening at UC Davis, and also to provide an opportunity to bring in guest external researchers who will also share their expertise. Our exciting lineup of monthly forums is scheduled through June 2021, and we've already received interest for participation in next year's series. Details can be found online at cannabis.ucdavis.edu under the Education tab. Next month's forum, titled Recent Advances in Hemp Genetics and Genomics, will take place on Thursday, March 11th from 11 to 12 noon, and will feature Dr. CJ Schwartz, Chief Science Officer of Sunrise Genetics. I hope you will join us for this and future installments, and tell your friends and colleagues. If you're not on our mailing list, you can provide your email in the Q&A, and we'll be sure to add it to the list. As today's presentation is underway, you'll be able to pose questions using the Zoom Q&A feature. Our presenters will save time at the end of the address, will have time at the end of the address to answer as many questions as time allows. This presentation is being recorded, and the recording will be made available in the next few days on the Cannabis and Hemp Research Center website, again, cannabis.ucdavis.edu under the Education tab. So let me now introduce today's speakers. We welcome Dr. Henning Langer, postdoctoral research fellow in the UC Davis Department of Physiology and Membrane Biology, along with Dr. Mark Mascal, UC Davis Professor of Chemistry. Henning and Mark were both separately recipients of our Research Investments in Cannabis and Hemp, or RICH seed grants that were offered by the center last year. Today, they'll be giving us an update of their research, generally, along the lines of the topic of the clinical potential of CBD and CBD analogs in neurological and neuromuscular disorders. So I'm not sure. Henning, are you going to start?
No, I'll start.
Perfect. Okay, so take it away, Mark.
Okay, so I'm going to share my screen, if that will allow me to do that. Yeah. And so can you see that? Everybody see that?
Yes.
Okay, great. I'm going to get the laser pointer there. Okay, so if everybody can see that, I'll go ahead and begin. So yeah, so I did have an intro page. There we are. And we've just been introduced, so I don't need that now. So we're going to talk about our approach to, and we've actually been, me and Henning have been working together as well. So we'll talk about some joint results, but I'll start with what we've been doing in this area. Okay, so these are the principal cannabinoids here, THC and CBD, which we're all totally aware of. And since we're going to be talking about these from a therapeutic perspective, that's what I'm going to focus on now. And the issue with THC is that it has a very narrow therapeutic window because it doesn't take much, only really milligrams for an adult to have debilitating cognitive and motor impairment. So your therapeutic window there is very narrow, and therefore the overwhelming number of studies of the pharmacological action of the cannabinoids has focused on CBD, which has a much lower degree of narcotic action, mainly soporific action. So there's been a lot of studies, and I'm going to talk about some of our interests in those potential medicinal indications. But one thing that has been overlooked to some extent, is the fact that it is trivial to convert CBD to THC. Okay, this is only a matter, for those of you who know chemistry, of just tickling this with a little bit of strong acid, which closes this down on here and gives you THC. And as CBD, pure CBD, becomes available in bulk, with its the decreasing regulations that we're seeing over the past years, there is a real danger of this conversion being carried out. As a matter of fact, it is being carried out. You can find papers in the literature. You can find recipes on the web. There have been arrests made. And the problem with this, you might be saying, well, what's the big problem? Because people can use cannabis recreationally. The real problem with this is that THC by itself, without any CBD to antagonize its neurological effects, it really is quite a dangerous drug. This is something that I started thinking about because I was very impressed with the therapeutic potential of the cannabinoids, but I was very worried about this conversion. So being an organic chemist, we got thinking about this, and it's pretty obvious that all you really need to do is take out this double bond. If you do that, you completely shut down the possibility of going to THC. So what we did was we made this H2 CBD, which is not natural. It is synthetic. It can be made from CBD, but it is not natural. So it's a synthetic, and you can have no conversion there. So this is, by any credible chemical pathway, this is totally impossible. And yet you've made such a subtle change to CBD that we thought that we would maintain the therapeutic properties. So that was the whole basis upon which we set out to do a program in this area. Okay, so how do we make H2 CBD? So it's very simple. We can take hemp-based CBD, and we can do what's called a hydrogenation. We hydrogenate out this double bond. This is a very simple, very clean, very straightforward reaction. And not only can we hydrogenate out this double bond, we can do this selectively, but we can also hydrogenate out this double bond as well if we want to. And that gives us another interesting analog, which we call H4 CBD, tetrahydrocannabidiol. Okay, and we've wanted to test both of these in a range of indications, but of course the first and foremost indication would be that's already FDA approved, and that is the mitigation of seizures. Okay, so we did a study of induced seizures in rats, and interestingly, we found virtually no difference in the mitigation of those convulsions between CBD, herbal CBD itself, and our synthetic H2 CBD, which is what you would expect. But of course, you can't just say that. You've got to prove it. And we did this study. We published it in this Nature Publishing Group Journal Scientific Reports in 2019. And this immediately got a lot of attention. So I just want to divert for a moment and talk about the altmetric score of a paper. This is, so of course you can look at the number of citations a paper gets, and that's an indication over the years, of course, of its impact. But the altmetric score has to do with how much attention it gets outside of the scientific community, sort of online attention. And a paper starts at 0, and most papers don't get much beyond 0, maybe 1 or 2, depending on if the authors are tweeters or not. Some papers, if they get a little bit of attention, might get into double digits. But for a paper to get much beyond 100 means that a lot of people are noticing it. And this one is nearly 300. So that puts it in the 99th percentile of papers of a similar age. So that's really pretty cool. So we got a lot of phone calls and a lot of people interested in this. We have a startup company based on this. So there's a lot of cool things happening here. Of course, the Rich grant that we got was also very helpful here in helping us to develop this this work further. So what are the advantages of synthetic H2CBD for this important indication, pediatric seizure indication that is currently the only really FDA approved use of CBD, although there are others in the pipeline, of course. The similarity stops here. Both of these are anticonvulsants, which we've proved in the animal trial. But CBD is still a controlled substance in many ways. Even though hemp-based CBD is less controlled than it once was, there's still transportation issues and other regulatory issues. And that's just talking about the US market. Okay, when we go to the global market, of course, things are much more restricted. So synthetic H2CBD is not formally a scheduled molecule. We've been on the DEA websites and we've gotten legal advice to tell us that we don't need a schedule anything license to work with this molecule. And that kind of takes me down here that synthetics should be able to penetrate into global markets where scheduled cannabis derivatives will always be prohibited. They'll always be stopped at the border, no matter how good they are, no matter how effective they are at seizure prevention. You're not going to get them in. Whereas this could just be marketed as a small molecule therapeutic that just happens to look like a cannabinoid. Okay, but that's our hope. We talked about this, the easy conversion to THC, that's abuse liability. There's no credible path to THC here. So that's a big advantage. CBD has sedative action. As far as we're aware, we see no narcotic action whatsoever in H2 CBD. So we dose rats up to the eyeballs with this stuff, and we see no changes in their behavior. And so there is some evidence in the literature that one of the reasons for the sedative action of CBD is that it converts to a small extent to THC in the gastric tract. Now, this is controversial. Some papers say, yes, this happens. Other papers say, no, it doesn't. But it sounds sensible to me because these, again, should behave pretty much the same. So maybe that's an issue that we're also avoiding here. Another major issue with CBD, at least for seizure indications, is that it has very low potency. So you have to dose this quite high, I think up to 25 milligrams per kilogram, which means that a person can take up to 1 gram of this a day to help control their seizures. This leads to hepatotoxicity issues, liver toxicity issues, leads to poor compliance because there are gastric issues there. So there's issues with the potency. And what we're hoping is that we can make analogs of this, further analogs of this, that have higher efficacy. All right, so how do we do that? Okay, that's a good question. The question here is, well, we can do this with the H2CBD that we make from CBD itself, but if we want to have a full sphere of possible derivatizations, so in all these positions indicated by the arrow, we can also do this synthetically using building blocks that are cannabinoid-like. So this is called philandrine. This is a natural product which you can buy. This is called olivitol. This is a natural product which you can buy. And if I play around with this philandrine, I can do some chemistry here. I can do some, for example, this is a tail group. It's a C5 chain in the cannabinoids, but I can play around with that. I can do all kinds of interesting things. Then I can put this together to get H2CBD analogs that are derivatized in these various positions. So this is an approach to making new analogs, and we are indeed doing that now and testing their efficacy. So the indications that we've been looking at, of course, the principal one has been epilepsy, and looking at not only H2 and H4 CBD, but some of the analogs that we've been talking about. This has been done, or is being done now in collaboration with Professor Mike Rogowski in the medical school. Of course, muscle recovery, which Henning is going to talk about in just a few minutes. We are also looking at metabolic syndrome, which is a collaboration with Rudy Ortiz at UC Merced. We're looking at slowing cognitive decline, which is a collaboration with John Voss here at UC Davis. Neuron regeneration, we're very early looking at that. We don't have really any data on that yet, but we're talking about doing that with my colleague in the chemistry department, David Olson. And we've given him the compounds, and we're hoping to see something interesting there. There's some evidence in literature that might be a matter of interest. Potassium channel modulation with Young-Fu Lin here at UC Davis, and then also pain with Karen Wagner, again, also here at UC Davis. So, of course, this is not all the things that CBD has been credited with potentially being able to do. Everything all the way up to heart disease and cancer and almost everything in between are things that people could look at. But these are the things that we are specifically looking at with our H2CBD analog. So I guess the key takeaways from this part of the presentation before I hand over to Henning is that we are developing a safer, potentially more effective, and more accessible treatment for seizures, and hopefully other indications that can unlock health care markets here in the US and certainly globally, where cannabis is either illegal or controlled. So I guess the idea is to rebrand and destigmatize the cannabinoid industry from the point of view of its therapeutic potential by the production of a non-narcotic and non-controversial form of CBD with no abuse potential across all of its possible markets. And since H2CBD and H4CBD themselves and some of their derivatives can be derived directly from hemp, success in this project will not negatively impact the hemp market. Now, there are some of the synthetic analogs which we don't make directly from CBD itself, but we're thinking that H2CBD and H4CBD are our lead compounds right now, and those are directly hemp derived. Okay, so I don't see any questions yet. I think those are saved for the end. And what I'll do now is stop sharing and hand over to Henning.
Thank you, Mark. Give me 1 second to share my screen. All right, I hope everybody can see this and hear me well.
Yes.
Okay, terrific. All right, thanks again, Mark, for your exciting introduction of these cool molecules that you are working on. Last time I had the opportunity to talk at this webinar, I briefly introduced the projects that we plan to run in respect to CBD and muscle or neuromuscular disorders. And today I will share some of the first results that we obtained in those projects. So part of what inspired us to work on neuromuscular disease and CBD is this cool paper that was published by a group in Italy in 2019 that showed that with CBD, you could ameliorate the symptoms of Duchenne muscular dystrophy in a mouse model for Duchenne. And what they saw was that they improved certain indicators on a histological level, but also cell differentiation in cell culture, maintenance of locomotor function in the mouse model itself, and decreased inflammation, as well as improved autophagy. So why are congenital muscular dystrophies an interesting target for CBD in general? And part of that is that even though congenital muscular dystrophies are by definition rare diseases, at least outside of Duchenne muscular dystrophy, they still sum up to a huge number of different diseases that are out there and that affect a relatively great number of people as part of the population. And currently there are very little treatments available for those people suffering from those several types of dystrophies. And despite that, they still share a number of pathological characteristics among them. For example, a lot of them go hand in hand with inflammation, with increased muscle protein synthesis or remodeling, and at the same time, an inability to efficiently process the proteins and break them down via autophagy. So there's an accumulation of basically proteins that are misfolded or aggregated, and that is something that CBD has shown to have some promise at. So we have recently, in collaboration with the mouse biology core facility at UC Davis, created a novel rat model for desmonopathy, which is another relatively common muscular dystrophy, with the technology of CRISPR-Cas9. And we have published that data in the Journal of Cachexia, Sarcopenia, and Muscle Diseases last year. And what we found is that those animals indeed show dystrophic signs, even if they are not challenged. But if we challenge them with a running exercise, you can see that the change in performance over 4 weeks is substantially greater in the mutant animals or even in the heterozygote animals. So you have this nice linear regression basically in performance between the wild type animals and the mutants in our desmonopathy model if we challenge them physically. So our question was, or what we plan to do as part of our grant application for RICH was that we wanted to see whether we can reproduce the effects of CBD in muscle cell culture in immortalized C2, C12 cells or in primary myoblasts of our desmonopathy animals. We also wanted to see whether CBD has any effect on exercise-induced muscle damage, because again, if we look at this data, there is a good chance that with exercise, we exacerbate the effects that we can already see at baseline in those animals, and that you see a greater effect of muscle atrophy and muscular dystrophy in those animals, while CBD potentially has some ability to ameliorate that, and the animals still get some of the benefits of exercising. And finally, we also wanted to see whether markers of muscle damage and inflammation would be modulated by CBD in those animals, but also in wild type animals. So to be more precise, the actual research questions that we came up to ask was, whether CBD has any effects on skeletal muscle after exercise in general, because that is a question that has not been answered yet, but which appears to be of high interest to a lot of people, giving the media attention that CBD receives in the context of exercise and regeneration. And another question that we wanted to answer in that context was whether CBD exerts any anti-inflammatory effects on skeletal muscle with or without exercise. And finally, if that is the case, that CBD has any anti-inflammatory effects on muscle, does it behave similarly to NSAIDs, which are also known to decrease inflammation, of course, but also have been shown to have some negative effects on skeletal muscle? For example, in this 2002 study from the Trapley lab, they shown that taking ibuprofen or acetaminophen after exercise decreases the muscle protein synthetic response to exercise compared to a placebo. So it appears to be that you need some of that inflammation or some of that inflammatory response after exercise to also reap some of the benefits of exercise. And another study, this time in animals as opposed to in vivo in humans, they've shown that if you do synergist ablation and you basically take out the gastrocnemius and the cilius in rats and then treat them with ibuprofen over 2 weeks, the increase in muscle mass that you usually see in the plantaris over the course of those 2 weeks in the control leg was substantially greater than the one in those animals that were treated with ibuprofen. So you have an efficient, almost entire blunting of the muscle growth that you usually see. So taking NSAIDs, yes, it does decrease inflammation, but it also hampers positive adaptations to exercise or to overload. And what we have been able to do in collaboration with Mark and our laboratory towards the end of last year was that we indeed tested CBD just in a basic proof of principle kind of manner and its effects on skeletal muscle after eccentric exercise in rats. I will briefly talk you through the protocol. So what we've done is we anesthetized the animals, and then we started eccentric exercise in those animals, which is essentially a stimulation of the sciatic nerve of those animals, which makes all muscles of a distal of the sciatic nerve contract at maximal intensity. And since the gastrocnemius and the cilius are more powerful dorsal flexors, the plantar flexor, they're more powerful plantar flexors. Dorsal flexor, tibialis anterior, experiences eccentric contractions because you still get the plantar flexion caused by the gastrocnemius and the cilius, but the TA works against that as powerfully as it can, and basically gets those lengthening contractions that we call eccentric contractions that are known to cause a disproportionate amount of muscle damage and stimulate muscle remodeling more so than regular concentric contractions. After the exercise, we injected the animals intraperitoneally with 100 milligrams per kilogram of CBD. So that's not quite as much as Mark usually uses for CBD and the H2 CBD to look at seizures, but it's still a decent dose compared to what is approved in humans for the treatment of seizures, at least. So it's if CBD does anything in skeletal muscle, we should be able to see it with that dosage. And then 18 hours later, we collected the tibialis anterior and other tissues to look at the effect of CBD. First of all, what we found is, and that is something that we've seen in that model that we use with the stimulation of the sciatic nerve across all our experiments, is that we see a slight increase in muscle weight on the next day in the leg that has been stimulated compared to the contralateral control leg within the same animals, which is probably mostly due to fluid redistribution, more blood flow towards the leg that has been exercised, maybe a little bit of swelling too. But if we looked at serum CK levels and the histology, then we really didn't see an excessive amount of muscle damage that has been caused in those healthy animals. So again, those are still healthy animals as part of this proof of principle test of CBD. And we did see that our intervention works in the sense of there was a decent exercise stimulus, but we didn't have excessive muscle damage either. And what we did next is, and what was our primary interest in those tissues that we collected, was to see whether any of the anabolic signaling or muscle remodeling signaling was impacted by CBD. And what we found there was that S6 kinase, which is part of the canonical mTORC1 anabolic signaling in skeletal muscle, basically a downstream target of mTOR, was increased in the vehicle, so in the control animals, as well as in the CBD animals to a similar extent. So S6 kinase, which is our, I would say, primary readout usually to assess muscle remodeling and muscle anabolism following exercise, was modulated to a similar degree by exercise, but not by the drug treatment. At the same time, another downstream target of S6 kinase, so that's ribosomal protein S6, the downstream target, the substrate of S6 kinase, was also increased in both groups, but showed a small tendency to be higher at baseline and with exercise in the CBD group. So that was interesting to see. And in terms of autophagy, which is another purported effect that CBD is supposed to have on certain tissues, we looked at ALK1, which is upstream of the autophagy pathway in skeletal muscle and most tissues. We didn't quite find a significant effect here, but you can see that there seems to be a little bit of something going on in the CBD group in terms of after stimulation having higher levels of ALK1. And similarly, P62, which I know is notoriously difficult to interpret as a modulator just on a sheer protein level, was also up in the CBD group at baseline as well as in the stimulated leg. Whereas LC3-2-1 or LC3-1 in general didn't really show a huge effect in those animals with CBD. Finally, we looked at cellular stress indicators on a protein level, and we found that muscle limb protein was up at baseline and with stimulation in the CBD animals, which is also indicative of more remodeling going on. We're not entirely confident in how to interpret muscle limb just yet. We know that it's upregulated in a lot of situations, but it is indicative usually of more remodeling in our hands. NF-kappa B, that was exciting to see, was slightly less stimulated with exercise, which might be indicative of less inflammation occurring in the muscle that was treated with CBD. And in terms of the chaperones, we really didn't see a conclusive effect of CBD because alpha B crystalline was slightly, or had a tendency to be slightly elevated in CBD across both groups, the control and the stimulated group. But heat shock protein 27, for example, seemed to be a little lower in the CBD group. So that was not entirely clear, but we were excited to see that the NF-kappa B looked like there was something going on. And in conclusion, we could summarize that there was at least no inhibitory effect on muscle anabolism of CBD, as opposed to what has been reported for the non-steroidal anti-inflammatory drugs like ibuprofen, etc. we saw a small decrease in local inflammatory signaling through the NF-kappa B pathway, a mixed effect on the chaperones, but overall very moderate to small effects of CBD on skeletal muscle after eccentric contractions in our rats. And to support that, more recently, I've conducted a bunch of cell culture experiments, not only testing CBD, but also those exciting molecules that Mark was just talking about, and those synthetic CBD analogs. And I will briefly run you through the experiment that I have been running, which is we took, again, C2, C12 cells. That's an immortalized mouse muscle cell line. I differentiated them for 4 days into full adult myotubes. And then I removed all the medium on those cells and I treated them for 15 minutes with PBS. And then half of the cells, I switched the medium to a growth medium that contained 20 nanomolar of IGF-1. And the other half of the cells continued for a full 60 minutes being starved on PBS, whereas again, the other half of the cells were incubated in the growth medium for 45 minutes. And what this does is that basically you can compare the protein levels in those starved cells, which will downregulate most of the anabolic signaling, to those that have been treated with the growth medium and the IGF-1, which is known to upregulate all those anabolic pathways that we have also researched after exercise. So saying the mTORC1 pathway with the P70S6 kinase, the ribosomal protein S6, et cetera, So it is basically mimicking to a certain extent exercise situation that that we have investigated in the rats. And of course, I tested CBD and H2 CBD on those cells, and not only the effect of IGF-1 on C2, C12 cells. And we chose 1 micromolar because in previous studies that has been reported to be a dose that has a certain effect on differentiation. Again, we have already fully differentiated samples here, but there is not a lot of literature on that subject. So we are really a little bit on the forefront here of that field, just trying those things out. But in the Italian paper that I mentioned earlier that showed that CBD had an effect in Duchenne muscular dystrophy mice, they also did a couple of cell experiments, and 1 micromolar seemed to have a little bit of an effect, whereas 3 micromolar already appeared to be toxic. So we went with 1 micromolar, and I looked at anabolic signaling in those cells, and what I found was exciting to me, maybe not quite as exciting as some would have hoped to, but I was happy to basically confirm the animal data that we collected. But the protein levels that we found for those major indicators of skeletal muscle anabolism were very similarly regulated to what we've seen after exercise. So that's IRS1, that's upstream of the mTOR pathway, and that is known to be, of course, stimulated by IGF-1 as well, because IGF-1 means insulin-like growth factor 1. And in the control cells, in the CBD cells, in the H2 CBD cells, across the board, the elevation of the cells that has been treated with IGF-1 was very similar. in S6 kinase, which is downstream of mTORC1 and the main canonical hub for the initiation of protein translation and muscle anabolism, was also very similarly elevated with the treatment of IGF-1 in the control group, in the CBD group, and in the H2 CBD group, with maybe a little bit of an interesting tendency here for the H2 CBD to be higher elevated than the CBD and the control group. And similarly, in the ribosomal protein S6 protein levels, again, that's downstream of S6 kinase, even a little bit closer to the actual initiation of protein synthesis in skeletal muscle, we've seen that control and CBD were on very similar levels, similar to what we've seen in the in our animal model, but also H2 CBD again showed a mild tendency to be further elevated than the CBD and the control group. So that was exciting, at least to me. And we can conclude that at least there is no inhibitory effect of canonical mTOR signaling through CBD or the CBD analogs after an acute growth stimulus through IGF-1. So that is confirmatory for our animal data that we have been fortunate enough to actually get approved and is going to be published soon in the International Journal of Sports Nutrition and Exercise Metabolism. But we still don't know whether those are the ideal dosages that we are using, whether longer term treatment of CBD has any distinct effects on skeletal muscle. We don't know whether inflammation is really reduced in skeletal muscle just on the basis of NF-kappa B signaling. That is a little difficult to tell. And we don't know whether those interesting molecules that Mark was talking about, the H2 CBD, the H4 CBD, and other analogs, might have an even more pronounced effect than CBD on skeletal muscle. To allow for an outlook and let you and share what we are planning to do in the future with our CBD project, one of the things that we definitely want to pursue in the near future is that we want to dial in a little bit more of the intricacies, how CBD works in vitro and how the CBD analogs work in vitro, and whether there are any effects that they might exert on skeletal muscle in a dish. That also pertains to the various dosages and the different durations of treatment that you can expose cells to. And then finally, we also want to, of course, test those analogs that Mark provided us with in a similar acute eccentric exercise loading scheme in the animals and directly compare them to CBD and a control group and see whether the analogs might have an even slightly more pronounced effect on skeletal muscle than the regular CBD has. Especially since CBD in itself had only a very mild or almost absent effect on skeletal muscle, even though the dosages were pretty high. So we have hopes that the H2 CBD is maybe a little more potent there. And then finally, also try this in our desmonopathy animal model, of course, acutely as well as after a chronic running exercise protocol, because some of the studies that are out there, especially on skeletal muscle, they suggest that maybe only chronic CBD treatment is really able to cause pronounced effect in skeletal muscle. And then finally, another relatively quickly experiment that we would be able to perform and that would be interesting to see is whether CBD or the CBD analogs might be able to sustain the muscle growth that you usually see with synergistic ablation. So that's what I showed earlier about ibuprofen blocking almost the entire growth of muscle after synergistic ablation, and see whether hypertrophy in the supraphysiological model, where you definitely have a lot of remodeling going on and a lot of pain that the animals are also exposed to, whether CBD has any modulatory effects there and is maybe at least superior to the NSAIDs that we've that have been reported to hamper the effects in the past, or maybe even improve hypertrophy. We don't know. So that's something that would be interesting to look at in the future. And finally, I would like to thank the Cannabis and Hemp Research Center of UC Davis to support us with the RICH grant to do some of this research that we have been carrying out here, as well as Mark and Keith Barr, my PI, for furthering the research that we have been pursuing there, as well as the lab members such as Suraj Pathak and Agata Mosakowski that were contributing to the research that I just showed you. And with this, I would be happy to take any questions now together with Mark, and I will stop sharing my screen. Thank you, everybody.
If you have any questions, please type them in the Q&A. At this time, there are no questions.
Let me ask a general question of
Yes.
maybe more for Mark, while people may think of their more detailed questions. So the doses that we're looking at here to treat neuromuscular disorders are orders of magnitude higher than the CBD doses that you typically see in various supplements that are readily available in the Davis community. And so I wondered if you could comment a little bit on that. I think it might be of interest to the participants here.
Sure. This is a function of the model. So if you have an animal model, if you want to get anywhere near the type of dosing that you would use in a human, you have to probably go an order of magnitude above that for a rat, because its metabolism is way faster than that of a human. So classically in these neurological studies, 200 milligrams per kilogram is perfectly normal dosing for a rat, and that would translate possibly to 20 milligrams per kilogram in a human. This is very approximate, but in terms of their respective metabolisms. So that's the reason that we do that. In mice, it's the same, if not even slightly faster than the metabolism of rats. So it just has to do with the animal model that you're using.
Yeah.
Good question.
So really good question, and maybe if I can add to that, I would fully agree with Mark that so we know rat metabolism and mouse metabolism even more so is faster than human metabolism. That's not only generally true, but also specifically for skeletal muscle. So we thought going with 100 milligram per kilogram, would be necessary to see any effects, if there were any. And even that still only caused mild effects in skeletal muscle, but still in the sense that there was a mild decrease on inflammatory signaling and a mild increase in anabolic signaling. So if we could get the best of both worlds, that would of course be exciting. But I would fully agree that 100 milligram per kg is still a relatively high dose with relatively moderate effects there, at least on skeletal muscle. But again, we don't know how it would look like in humans. And I will also say, this goes back to what Mark was saying earlier about the potential conversion of CBD to THC in vivo, that some of the animals did look indeed not only sedentary, but a little drowsy and a little challenged with the CBD after 18 hours. And that there has been, when we open up the animals, of course, but while collecting the tissue, also some gastric intestinal issues with them. So I wouldn't be, I'm not an expert, of course, but I wouldn't exclude the possibility that Mark was mentioning earlier, that the conversion of CBD to THC is mainly run through acidification, and that of course in the gastrointestinal tract there is some acid that I don't know in to which extent this would be able to contribute to that. But apparently there are studies that have found something like that, and that is somewhat in line what we've seen in our animals after 18 hours. And it would be interesting to see whether we could circumvent that by using the analogs, which cannot be converted back to THC. So maybe there is an even clearer effect of the H2 CBD than there is of the CBD in muscle. But there are, I see a couple of the Q&As there. One more was also targeting the dosage. And I've been going through this quite a lot in the literature, especially as part of our animal ethics application. And there is a wide range of what people have tested in rats, and they go up to 200 milligram per kilogram and more in rats daily for weeks and weeks without problems. Again, we have seen at least smaller problems already at 100 milligram per kilogram after a one-time acute dose. So it's also I don't know how clear the literature really is on, what impact the animal model has, different rat strains, different sex of the animals, all these kind of things. I don't think it's very well defined yet. And at some point, you just have to go with one dose that seems reasonable based on the available literature. But I think there will still be a lot of work going into that going forward, dialing in the ideal dose of CBD.
Yeah, and these are good questions here that are basically what Cameron just asked. But the important thing to note is that the human trials have shown that you get about the same effectiveness at 20 milligrams per kilogram in a human that you get with 200 milligrams per kilogram in a rat. So if I dose a rat at 20 milligrams per kilogram, I don't see anything, nothing whatsoever. But if you couldn't dose a human at 200 milligrams per kilogram, so there really is experimentally defined differentiation between these, and it seems to be at about an order of magnitude. Off-target effects of H2CBD. It seems to operate. I mean, we haven't looked at this in huge detail for Jonathan's question. We haven't looked at this in massive detail, but as far as we can see, and this is probably to be expected from a molecular basis, is that they seem to be behaving pretty much the same, except for that conversion to potential conversion to THC. For everything that we've looked at so far, they seem to behave pretty much the same. Now, time will tell whether there's any specific differences, maybe with some targets and some indications, but so far, what we see is pretty much identical mode of action.
Well, thank you both. Let's give a brief minute for any last minute questions. Coming back to my original question, most supplements that you buy, that one could buy with CBD, have maybe 5 milligrams, 20 milligrams at most.
Yep.
So that was really, I think, part of my question is what is the therapeutic window for CBD,
Yeah.
and what effects would you predict with doses in that low range?
That is a really good question. Sorry, Mark. Let me go ahead real quickly.
Sure.
Just on the muscle side of things. Well, 5 milligrams is what you see in a lot of supplements. You were just saying, Cameron, but I think that even specifically topical treatment, you always lose a little bit of the efficiency when you go from injection to even oral intake of a drug to topical treatment. And then if we take that into account, plus the fact that we have such a higher dosage to what has been suggested by retailers or supplement providers, I would be surprised if those tiny dosages that are available have any measurable effect on skeletal muscle beyond the placebo effect at this point.
Yeah, absolutely. You are basically throwing your money away if you buy those supplements, because they have tiny quantities of CBD. Comparatively speaking, the number of mgs per kg would be in the noise. So I think that any effect that you're getting there is a placebo effect. If you feel better after you take it, that's the only thing that could explain it, because it's way beyond a physiologically meaningful dose, those supplements. And indeed, since they're not controlled, you don't even know how good
what exactly is in there.
Right. Yeah. You don't know what's in there and whether the CBD is pure. And so I would strongly recommend anybody listening to not waste their money on that.
Yeah. And I will maybe add that what we looked at here is skeletal muscle, and in terms of Mark, he has a lot of collaborations, but the paper that he presented was on seizures. If people feel that it has an effect on sleep quality or anything like that, that's something I haven't looked at with my data, and that I'm not very well versed in terms of knowing the literature on that. So I don't know whether CBD at smaller dosages might have any effects there. But in terms of topical treatment, I would highly doubt that. And even if you take it orally in those small dosages, I would be very surprised if that does anything to your body.
Very unlikely.
So we have one last question from Randy Hageman about effects of CBD on mitochondrial function.
Good question. We haven't looked at that yet. That is definitely something that we will also explore going forward as part of our broader characterization of the effects of CBD on skeletal muscle, but we don't know. And none of the literature out there is really suggesting any clear effect there. And I wouldn't know how CBD via its receptor action on skeletal muscle would exert any effects on mitochondria. But again, that's an entirely open research field that nobody has really tapped in so far. So it's a really good question, but I unfortunately have no answer to it.
All right. Well, I think that is our last question, and I think we're
No,
at
there's
the-
one more question, Cam. Sorry to interrupt.
Good.
It's from
Sorry.
Gino Cortopassi. The question is, Mark, the usual human dose of CBD that is given to humans is about 20 milligrams per human per day. What dose of CBD is therapeutically effective?
So yeah, that seems to be a therapeutically effective dose for the seizure indication. That's for sure. That's about right. And so you're talking about, depending on how many kilograms you are, you're taking hundreds of milligrams potentially a day, usually over 2 doses, spread out over the day. Now it may be in other indications that you can get away with less than that. I think that's, but yes, what you've written is correct, and that's again another point about why these commercial tinctures and things like that are simply not going to do anything meaningful. I hope that answers the question.
Great. Well, thank you. Thank our speakers. Let me remind you that we have another forum next month, and that will feature Dr. CHRC, Chief Science Officer of Sunrise Genetics, talking about hemp genetics and genomics. And that will occur on March the 11th at this time from 11 to 12. So I'd like to thank our speakers and our attendees for their attention and questions. And we look forward to seeing you all next month.
Thank you very much.
Thank you.